We'll now turn our program over to Steve. Thank you, Steve.
Thank you, Diane, and thank you, Jean. Jean is going to keep an eye on the chat, so if you have questions, you might just drop them into chat. And I plan to break a little more than halfway through the talk. I want to say right off that this talk is likely to run more than an hour. I've tried to shorten it, but some of you have had maybe even 2 of my DNA courses and are familiar with a lot of the background material, but I think a lot of you are taking this for the first time, so I don't want to rush ahead. So we'll go over a lot of the basics right at the beginning. Let me just start with the screen now and show some slides. Takes a minute. If I can move ahead on this. Always takes a second to get the slides moving. Okay, so my title, which I believe you probably all read, is Decoding the Genome, the History and Science Behind the Human Genome Project. And this is really one of my favorite subjects. This took place, it began in the 1980s. People started discussing it. And it officially began through the auspices of the NIH. The director's office set up a special office just to work on the Genome Project in collaboration with the Department of Energy, which historically has a role in this sort of work. That began in 1990. It became an international effort with a huge contribution from the British lab and the British people working in a place called the Sanger Center. We'll come to the Sanger, the man behind all of this, and other labs from around the world participated, but U.S. labs, several U.S. labs, 4 of them in fact, played a major role in this, including our own Broad Institute in Cambridge. So it was a large project. I'm not really going to tell the story behind the politics. What I want to focus on today is kind of the exploratory nature of the Genome Project and the role of basic science in giving us a handle on many of the puzzles that the genome presents. So to get this moving, I take this page from the Human Genome Project at the National Human Genome Research Institute, which is founded during the Human Genome Project. And they talk about this project as being one of the greatest feats of scientific discovery in history, a reasonable thing for them to say. And they go on to characterize it as a voyage of scientific exploration. And I like that because when they started, they did not really know where it was going to take them. They didn't have the technology to do it really, but it all worked out. It was deemed complete in April 2003, where the Human Genome Project gave us the ability for the first time to read nature's complete genetic blueprint for building a human being. There's a little bit of overstatement here, but I think that's a fair description overall. There are a couple things that inspired me to do this topic today. And one of them is that the actual completion of the human genome, a really complete sequence of the human genome, actually was published. The last part of it was published last summer. So it's not something that happened 20 years ago. It's something that is still happening in a sense. And the other thing is, many of you may have seen this. It was in the papers extensively a couple months ago. A new technique for editing genes has actually found clinical application and has been approved by the FDA. And we'll talk a little bit about that too. So these are some of the final things I want to talk about.
Pretty good, you got it.
Okay. So what is the human genome? Well, the term genome is actually a pretty recent term. It simply refers to the collection of all the DNA in a typical cell of a given organism. And so here we have a rather elaborate visualization of the human genome. This is a technique for staining DNA that's very fancy. I can't think of any other word, where we see the chromosomes of a human being all splayed out. This is what chromosomes look like just before a cell is getting ready to divide. And there are 46 chromosomes in all here. They're arrayed in what's known as a karyotype, from largest to smallest down here. There are 23 pairs, 46 chromosomes. 23, one set from your mother, one set from your father, forming pairs. The cell is about ready to divide. So each of these elongated bodies contains 2 strands of DNA, one strand for each of the new cells. Up here, we see the same staining of a cell nucleus, this globe, what might look like a map of Middle Earth, perhaps. This globe is actually the same type of staining technique, only now the DNA is dispersed in the undividing cell. And so you see patches of color, but the DNA is spread throughout the cell nucleus. So I say it's the collection of DNA in a typical cell. What is DNA? And I want to focus on that topic for the beginning of the talk. DNA is a long, stringy molecule, and this is one way of looking at it, where it's not really stringy at all. This is a light microscope depiction of the human genome. But if we go to a much finer level of resolution, like this, we can actually see DNA as a molecule. And this is a bacterial cell that's been broken open in such a way that the shell of the cell is still intact, but the DNA has leaked out and formed this long halo. And I want to give you some idea of DNA as a really long thread-like molecule. If you were able to measure this DNA, all the loops and whorls that I show in this picture, you would find that in real life, we're looking at about a little less than 2 millimeters of DNA. That's less than 1/16 of an inch of DNA. So it gives you an idea how thin this DNA is, because the entire length that you're looking at here is very, very small. The human genome, if you could lay all the DNA in those 46 chromosomes end to end and stretch it out, would measure about 2 meters. And so you have trillions of cells in your body. So when you think about this, trillions of cells times 2 meters, 30 or 40 trillion cells, you actually have in your body, each of us has something on the order of 40 or 50 billion miles of DNA in your body. Of course, it's far too microscopic to see or to stretch out, but we're talking astronomical distances that are far larger than the distance across our solar system. So DNA is a very long, very, very thin molecule. A lot to learn here. Moving down one level further to look at the actual atomic level of DNA, we can think of DNA as being a chain-like molecule. A thread is maybe a little too smooth. It's broken up into units, into links. And each link in this chain has 3 parts: a phosphate group, which is acidic. So the term DNA refers to an acid. The A is for acid. It has a sugar. The sugar is called deoxyribose. So that's the D in DNA. And then it's in the nucleus. So that's where the nucleic part comes from. So DNA is deoxyribonucleic acid. And what's distinctive about it is that each of these links has one of 4 bases attached. The term bases is very general, but that's just a term for describing one of these complex molecules. They're rather large, kind of flat organic molecules with carbon, oxygen, and nitrogen in them. And these are arrayed in the following way. The phosphate sugars form the backbone. So it goes phosphate, sugar, phosphate, sugar, phosphate, sugar. So you have a very repetitive structure here, but off of each sugar hangs one of the four bases. And this is what distinguishes the DNA. And when we talk about the sequence of DNA, we're really talking about the organization of these four bases, whether it's an A, C, G, or T. Now, another way of looking at DNA is to prepare it in the laboratory. And this is a highly purified sample of DNA, but it's actually where the key investigations, the biophysical investigations of DNA began back in the 1930s and 40s. People were able to draw out fibers of DNA like this from a water alcohol solution, as you see this person here pulling out the DNA, sort of a snotty looking substance. But as you draw it out in this type of fiber, the DNA filaments at the molecular level, the DNA filaments arrange themselves parallel to the axis of this drawn fiber. And this gives the fiber some degree of molecular structure. And so you can study this by putting it in an X-ray beam. And this shows an early X-ray diffraction pattern of a fiber. And it's pretty blurry, but there's some structure here. And the position of these dark lines here actually is indicative of a intrinsic repeat within the structure, not just of the chemical structure, but within the way the atoms within that chain are arrayed in space. And so this type of data has been known for some time, but in the early 1950s, and I think in the year 1950, a biophysicist named Maurice Wilkins, working in London, just started to investigate the structure of DNA again. And he had a much higher quality sample, not unlike the one that I just showed you, being drawn out of alcohol. And he got this supply from a friend of his in Switzerland, so it was very, very valuable. And he quickly found a way to pull out the fibers and put them in an X-ray apparatus, put them in an X-ray beam, and then look at the way in which the beam, a small amount of the beam would be scattered from the fibers. And he got a better picture than the one that was published. And his boss immediately recognized that this was a significant advance, that things were primed for some discovery. And so his boss went out a little bit behind his back and hired a specialist who had been working in France, Rosalind Franklin, who came to the lab in early 1951. And working pretty intensely, she refined Wilkins' methods. She also took his graduate student to help her with it. And they got a very nice picture in early 1952 that had considerable more detail. And this type of pattern has to be analyzed by mathematics. It's a very demanding sort of thing, especially back in the early 50s when computers were not readily available. But they were able to make certain conclusions as to repeat distances. One is noted here on the atomic scale, and say something about the symmetry of the molecules that lay within this fiber space. Wilkins went off to a meeting in Italy and gave a talk, showed some of his diffraction data, and immediately caught the attention of a young American named James Watson. I think many, if not most of you, have heard of Watson. He was a child prodigy, got his PhD when he was 22, and now he was 23 and had come to Europe to do some postdoctoral work. And when he saw that this new data on DNA, he immediately changed his plans for study and managed to land himself a position in one of the most famous physics labs in the world, the Cavendish lab at Cambridge University. And so when he went there in 51, he met this older student, a 35-year-old Francis Crick, who was still trying to finish up his PhD, having been interrupted by the war. So these two people, about 12 years apart in age, hit it off. And the rest is history, because their discovery, based in part on the work of Rosalind Franklin, but considerable insight on their own, allowed them within the space of a little more than a year to assemble a model of the three-dimensional structure of DNA, not just the chemical structure, which had actually been worked out in its final details a year or two before, also at Cambridge University. So this was a very good place to be working on DNA. And this model kind of very neatly fit Rosalind Franklin's data, which was not so curious, since they had used the data to determine some of the key parameters of the model. So now I want to sort of show you this model in more detail than you can see in this old photograph on the right. This is one of my favorite pictures for showing the structure of DNA, because it shows it in two different modes. On the far left here is the ladder structure of DNA, where the phosphate sugar backbone is shown as two rails of the ladder. So there are two strands of DNA in this DNA molecule, giving rise to what's known as the double helical structure. So one strand of DNA, one phosphate sugar strand, runs in one direction, and the other one runs in another direction. You don't normally think about the direction of the rails of your ladder, but this is what we call an anti-parallel configuration, and it has some important implications for DNA activity. And most importantly, and what the real insight that sort of put this model together is the role of the bases, because the bases are sort of hanging off of the phosphate sugar strand, the phosphate sugar backbone, and rather than just sort of hanging out, protruding from the strand, the bases on these two strands that run parallel to each other kind of join up using these sort of quasi-stable chemical bonds known as hydrogen bonds. So here we have the A base here that can line up very neatly with the T base and form these dotted lines, these so-called hydrogen bonds that sort of link them together. And similarly, the G and C bases line up. So we have one large base and one small base in each of the pairs. And these are flat molecules, as I may have mentioned. And so they stack on top of one another within the ladder, sort of like rungs on a ladder, but they actually, at the molecular level, they actually sit one on top of another. And this accounts for the major spacing that the fiber diffraction patterns that I just showed you have, the 3.4 spacing. And so this means that there are, in that 6 feet of DNA that I described, just calculating on the basis of this repeat distance that running down the double helix, we can calculate that there are roughly 3 billion base pairs of DNA. And that's a lot. That's a lot of letters, a lot of A's, a lot of T's, a lot of C's, and so forth. But going back to the ladder here, we see that wherever we have a G on one strand, we have a C on the opposite strand. And conversely, if we have a C on the first strand, we'll have a G opposite it. A T here leads to an A there, an A there leads to a T. So these pairs have what they call pseudo-symmetry. That is, you can flip the pairs around and accommodate all 4 organizations of the 2 pairs within this uniform structure. And so in this space filling model here, where the atoms are actually shown as spheres, we see the 2 strands in this what looks like mostly purple and white running parallel to each other, one strand and the other strand here. And we have a big group here where you can see the edge of the base pairs. That is the edge of this one here, or the edge of the GC pair shown in orange. And on the far side, we see just a little peak of the yellow side of these base pairs. So this is the double helix structure, which has become famous. And it was this model that actually allowed people to see DNA as the genetic material. In 1953, biochemists around the world, or rather, and biologists around the world who thought about the nature of genetics at the molecular level, were really pretty divided. A lot more was known about the structure of proteins, a molecule that I'm going to talk about very shortly. Proteins seem to have more diversity and carry out many, many functions within the cell. And people tended to think of DNA as being the structural component of chromosomes. They were aware that chromosomes contain both DNA, but also a lot of protein. This model actually showed the reason why DNA really was, and supporting results of other people's observations, DNA was the genetic, the molecule of genetics. And I want to just, there are two ways that this model fits that concept so clearly. One way is by having two strands in a given DNA molecule, that each strand has the same information. It's just in one strand is complementary to the other. So these two strands can separate and serve as templates for the other strand. The other thing is that this uniform structure can obviously accommodate any sort of sequence. So in the sequence of these bases, these four bases, you can actually store a lot of information. So you can think of this as some sort of molecular computer tape that can just be read off. So we can think about these ideas as two problems. One is how is genetic information duplicated? And we refer to this as the replication problem. And the answer to this was intuitively obvious, and Watson and Crick published a second paper very quickly after their first publication of the model, showing how they thought the strands might separate. Each strand would serve as a template, so that as the cell divided, there would be two copies of the DNA molecule. Each copy would have one strand of old DNA and one strand of new DNA. And this model for replication was supported at the biochemical level very quickly. But what I want to focus on today is I want to focus on the other problem. How does information encoded in the sequence of DNA direct cell function? That is, at this point, genetics was a fairly well-developed subject. People have been studying genetics in fruit flies, in corn, even in bacteria, and people had begun to study genetics in humans too. They had the idea that certain diseases were inherited within families, and so the idea is that the gene codes for information, and somehow this information is realized in the characteristics of the cell or the organism. And not only that, there had been a lot of work done on protein chemistry, and proteins were now beginning to be recognized as long polymers too. When I say long, it's on a different scale than the length of DNA. Proteins represent short, long polymers made up of different kinds of repeats, amino acids rather than the bases that I've been telling you about here. So that suggests the problem of, well, how does the sequence of bases in DNA translate into the sequence of amino acids in proteins? And this is the coding problem. And whereas the replication problem seemed to have a fairly straightforward solution, at least in principle, the details can get quite complex when you get into the biochemical and genetic weeds. But the idea here that is shown at the top is a fairly simple idea. But how to take 4 bases and come up with the structure of amino acids and proteins was not an obvious problem, and it took people more than 10 years to work it out in detail and even really in principle, and that's really what I want to focus the this part of the talk on. And there are 2 people that I want to introduce you to that both contributed to this in a major way, and both had to do with the structure of proteins, also working at Cambridge. And this work actually historically overlapped with the work of Wilkins, Franklin, Watson, and Crick on DNA, because he began a little bit earlier in 1949, and he was still working on it as they were putting the double helix together. But by 1953, in another part of Cambridge University, in a chemistry lab, a man named Frederick Sanger was working out the detailed structure of a very small protein, insulin. And of course, insulin is an important hormone that controls the level of blood sugar. Many diabetics rely on insulin to help insulin injections to maintain a level of a physiological level of glucose in their blood, and so this use of it as a therapeutic agent meant that there were relatively pure samples of insulin available, and something was known of its size, whereas the size of many proteins and the purity of many proteins at that time was suspect. But Sanger's work moved fairly steadily, but it took them 5 years to work out the complete sequence of amino acids in what turned out to be 2 chains in the insulin molecule, with a total length of about 50 amino acid residues, and it's shown at the bottom. I'm not going to say much about it, but there are 20 amino acids that are commonly found in proteins, and here you see a sequence with a specific order of almost 20 different amino acids in it. And this work, this careful chemical work, showed for the first time that proteins were actually had a defined sequence, specific sequence. Otherwise, he wouldn't have been able to come up with a unique sequence. And these were things that people were uncertain about, but it fit very nicely into the idea if DNA, that DNA might have a fixed sequence of bases, some of which would specify the fixed sequence of amino acids. But how? Adding to this story was another world famous chemist named Linus Pauling, who had been studied in Oregon and done his PhD in Caltech, and spent some years in Europe establishing himself as perhaps the outstanding authority on the nature of chemical bonds. And in the 20s and 30s, Pauling's work on chemical bonds and in all sorts of molecules really sort of set the stage for modern chemistry. And in the late 1930s, Pauling turned his interest to biomolecules. And in 1949, he published a whole series of papers. In 1951, he published a whole series of papers describing fundamental features of protein structure. And this was at a time before anyone had any idea of what sort of ways amino acids could arrange themselves. With Pauling just knowing the atomic details of how the details of how atoms in proteins are arranged and how these chains of amino acids could fold, could come up with a number of structures that turned out to be almost prophetic in their nature. And it was Pauling's success with this type of molecular model building, sort of showing Pauling pointing to his famous alpha helix, that inspired Watson and Crick. They were both extremely familiar and impressed with Pauling's advances in molecular biology and sort of their adherence to model building as a way to look at DNA structure really came from Pauling's work. But Pauling had already carried out another famous experiment, and it was actually a very simple experiment. And it was in a completely different area of protein chemistry, where a disease called sickle cell disease or sickle cell anemia had been identified at the beginning of the 20th century. And what would distinguish it is that normally, here's a US postage stamp showing red blood cells in the background around Pauling. The normal red blood cell is sort of a disc-shaped shell, a hollowed out disc. And in sickle cell disease, blood cells are distorted and take on this so-called sickle shape. And these sickle shells are unstable. They block capillary vessels of the process, something known as vascular occlusion crises, affect people with the sickle cell disease. It's very painful. It damages organs, livers, and kidneys, and often shortens lifespan. It's sort of a variable disease where they can be normal some of the time, but never not for long. And Pauling realized that red blood cells are, in a sense, nothing more than a membrane sac filled with hemoglobin, a very dense, almost paste-like amount of hemoglobin. And this hemoglobin within your red blood cells is what binds oxygen in your lungs, picks up the oxygen in your lungs and delivers it to the cells in your tissues. And to do so, of course, it flows from large arteries into small capillaries. And that's where the sickling causes the great damage. And I want to talk about this a little because sickle cell disease turns out to be an important application of human genomics. So in order to talk about hemoglobin, I want to just introduce you to a little bit of more protein structure. I said proteins are chains of amino acids, very much the same way that DNA is a chain of bases and phosphates and sugars. And but the chains are much shorter. They're on the order of 100 to 3,000 amino acids, not millions of them. And these chains, because there are 20 different amino acids, the chemistry of these chains is much more diverse. And so they can fold up in many, many different ways. They're not just a uniform structure at all. And this just shows some of the diversity. It's also a little glossary of the different functions that proteins play in your body. Collagen is the most abundant protein in our body. It's the proteins that hold our bones and our cartilage together and provide the strength and flexibility of both. So it's a structural function. Proteins are the most abundant substance, taken collectively, the most abundant substance in your cells, aside from water, which makes up most of the mass of your cells. We're mostly water. But taking the water aside, most of your body is made up of proteins, and they come in many varieties. Antibodies are proteins that are found in the blood that help fight infections, bind to specific invading substances. Hemoglobin is, as I said, found in the red blood cells. So these are both blood proteins. I'm going to come back to hemoglobin. Proteins are found in your membranes. Your cells are surrounded by membranes. They're membranes inside of your cells. And so a major constituent of the membranes are proteins, and many of the proteins play very specific functions in communicating across membranes. This is a membrane receptor, so a hormone in your bloodstream might bind to this membrane restructure and trigger a response inside the cell. Thousands of proteins within your cell are called catalysts. That these are proteins that direct and speed up chemical reactions in your body. So your digestive system is full of enzymes that digest proteins and carbohydrates and all the other substances that you consume and use as a source of energy. And finally, proteins interact with DNA. And so here we have showing how proteins and DNA bind to one another, a double helix here in the red and blue, and a protein here. And most of these depictions, with the exception of collagen, are what we call ribbon diagrams. And these are actually not showing you the full bulk of the proteins, but rather these are just focused on the backbone. And so all these helices show you the helices that Pauling first noted as a feature of protein structure. And other of these diagrams show you other ways in which the protein backbone can be arranged into sheets with these ribbon-like structures that Pauling also predicted. So here we have DNA and proteins entering into our story, beginning to get some sense of how these two linear molecules, these chains, might be related to one another. I just want to say a little bit more about hemoglobin function. I've mentioned that hemoglobin carries oxygen in our blood. So we see oxygen molecules up here binding to sites within the 4 chains of hemoglobin. So hemoglobin, like many proteins, is made up of multiple amino acid chains, and hemoglobin is made up of what we call alpha chains and beta chains, and there are 2 copies of each. And when oxygen binds, this combination of alpha and beta chains rotates with respect to the other alpha beta dimer. And so there's this very subtle change in hemoglobin structure. And that's relevant to sickle cell disease, because the sickle cell hemoglobin that Pauling first characterized, and in his characterization, Pauling showed that the hemoglobin in sickle cell disease is different than the hemoglobin in normal humans, and that there are other people that have what we call sickle cell trait, a mild form of the disease, which have copies of both the normal hemoglobin and the sickle cell hemoglobin. And Pauling's work on sickle cell hemoglobin was correctly predicted that there would be just a small number of changes in the larger hemoglobin molecule, maybe 2 changes in the hemoglobin molecule. And that prediction proved to be right on. I think he said 1 to 3 changes in the hemoglobin molecule. And it turns out that that represents a single change in amino acids within the beta chain of hemoglobin. So 1 change of 1 amino acid and the 2 copies of beta globin lead to the sickle cell form of hemoglobin. And when it is in the deoxy form, that mutated, altered globin forms these long fibers. And it's the formation of these fibers in the deoxy form of hemoglobin, that is hemoglobin that is unloaded its oxygen, that causes cells to sickle. So Pauling's demonstration of sickle cell hemoglobin, which came long before, years before people knew what the structure of hemoglobin looked like, correctly predicted that a small change in the amino acids sequence could lead to a molecular disease, and that this change was inherited from generation to generation. And so this kind of complemented Sanger's work on protein structure and showed a very clear relationship between inherited information and the structure of proteins. So that's why I'm spending so much time with Pauling's insight here. So how did this translate into connecting the sequence in DNA with the sequence of amino acids in proteins? And the breakthrough here came through the observation of the site of protein biosynthesis in cells. And it is particularly clear in animal cells, where you have a cell nucleus, as most higher organisms do. You have DNA located in the nucleus, and the protein biosynthesis was located in the cytoplasm using radioactive amino acids in the late 40s and early 50s. Again, this work is sort of simultaneous with some of the work on DNA and proteins, and it was found to be located in these little granular bodies that were both in the cytoplasm of cells, that is the contents of the cell outside of the cell nucleus, but also attached to these internal cell membranes known as the endoplasmic reticulum, or just ER for short. And these granular bodies turned out to be the site of most of the RNA in a cell. So these bodies were named ribosomes or RNA bodies. And an RNA, as its name kind of suggests, is a close relative of DNA. The RNA differs from DNA only in the nature of the sugar, which is a closely related sugar called ribose, as opposed to deoxyribose in DNA. So it's the difference here is one more oxygen atom than the deoxyribose has. And it has a slightly different base composition. It has what we call U instead of T. T and U are very similar. So U, like T, pairs with A. So the idea of the RNA is that it's a very close cousin of DNA, except it occurs primarily as a single-stranded molecule. There can be double-stranded forms of RNA, and short stretches of RNA form a double helix, but RNA is usually seen as a single-stranded molecule in the cell. And RNA turns out to play a major role in protein biosynthesis, but also in many other aspects of the cell. So I want to kind of put a big star around RNA, because that's going to be the third major player in my talk about the genome. This is just an historic electron micrograph showing a section of a cell where we have the nucleus on the right here, and we have a little bit of the outside plasma, the cell membrane here. And what we see here is a large amount of endoplasmic reticulum. And you can see very clearly in this electron micrograph, you can actually see the granular ribosomes. This is a pancreas cell, it's a secretory cell, and it's making huge amounts of a digestive enzyme, which are being secreted from the cell and dumped into intestinal tract through the pancreatic ducts. So it's this type of examination and characterization of the ribosome that allowed people to understand the role that ribosomes play in the biosynthesis of proteins. So for a long time, people assumed that the RNA within the ribosome itself was what was responsible for proteins, and it wasn't at all clear about how this worked. But in late 1960, two labs, one led by James Watson, the other lab in Paris, representing a consortium of many genes, showed that there are small transiently expressed RNA molecules known as messenger RNA molecules that in animal cells would be synthesized in the nucleus where the DNA is, and then transported out to the cytoplasm where the ribosomes are, and that these molecules associate with the ribosomes and actually carry a sequence of segments of the DNA out to the ribosomes where they direct the synthesis of protein. So I have a couple slides here just outlining this key process. This represents the DNA in the chromosome, so the DNA template. One of the strands of DNA here is copied into a single strand of RNA, and then that strand of RNA is transported out of the cell nucleus. Bacteria don't have cell nucleuses. A lot of this early work on messenger RNA was actually done in bacteria, but it's somewhat clearer in the nucleated cell of humans and other animals that the DNA resides in the nucleus. The mRNA is synthesized there, but is quickly transported out of the nucleus and associates with the ribosome, which serves as a reading device. So most of the RNA in a ribosome is actually part of the reading apparatus that assembles amino acids based on the sequence of bases in the messenger RNA. So this type of diagram where DNA codes for RNA and the RNA somehow codes for proteins was intuited around the late 1950s by Crick, who published papers and gave lectures on this subject. It was there was a lot of data to favor it, but it wasn't until a young American biochemist, relatively unknown by the labs in Paris and England or in Cambridge or California, a man named Marshall Nirenberg, working at the NIH, was able to prepare a preparation of ribosomes and show that a synthetic form of RNA that he could make that contained just the U base, so it had a sequence of U's. He was able to show that he could program the ribosomes to read the sequence of the U's and incorporate a single amino acid, and that amino acid was called phenylalanine, and that was the beginning of actually decoding the genetic code. And at the bottom here, I show the genetic code book. I don't want to go over this in any detail, but basically what is being read are 3 bases at a time, and 4 bases, that is the 4 bases, A, C, G, and U, can be arranged into 64 triplets, and each of those triplets codes for one of the 20 amino acids. So some triplets code for multiple amino acids, and there are actually three triplets that don't code for any amino acids, and these turn out to be the stop signals, the ones that tell the ribosome that it's reached the end of the message and that it has completed the protein. So Nirenberg's lab sort of won out the competition between a number of better staffed labs that had already established a reputation in the field of molecular genetics. His unit at the NIH did a tremendous amount of work over the course of just about 5 years to assemble most of the code book. But there were other people in other labs working on this at the very same time. Nonetheless, a few years later, he was awarded a Nobel Prize, and his lab celebrated. So this is a picture of the Nuremberg lab welcoming Marshall on the day that he was awarded the Nobel Prize. So, and with the first code word of the code dictionary shown above. So now I've sort of taken you through the basics of molecular biology. I want to stop here and see if anyone has any comments or questions about what I've covered so far. How are we all doing?
There was one question on your first slide, Steve, about it showed the X chromosome, but it didn't show a Y chromosome, and the person was wondering, where's the Y?
Yeah, that's I appreciate that question. The Y chromosome, what we were looking at there is a female karyotype. So that was a cell taken from a woman, and it has 2 X chromosomes. That's what distinguishes men and women. The Y chromosome is, of course, found exclusively in males. And as a male, I can say it's a fairly miserable little chromosome that I think it may be smaller than the next smallest chromosome. And it was also the hardest one to sequence. And it was the sequencing of a single Y chromosome that actually completed the human genome last summer. And so it's worth taking note of. And it was a difficult one because it contains a lot of so-called junk DNA that no one really believes is junk anymore, but we're still a little puzzled about how it all fits together at that level. And any other questions?
You have a few hands have gone up. Paul?
Sure. I was like to try to get a little more out of Steve. So I noticed in your last chart, Steve, there seems to be, all these doubling and triplings of things, presumably for redundancy to not make mistakes in making new chemicals. But tryptophan only appeared once.
Oh, well, yes.
I'm wondering if that has some significance, like there's supposed to be a mutation somewhere down the road that makes 2 copies of tryptophan, or was that just an accident? What's going on?
Yeah, no, I mean, this is sort of nature's own code book. And we honestly don't know where it came from. I mean, there's a lot of speculation. I've spent some time reading some of this, and there's mathematical analyses of the code that suggests that it's a robust code in some ways. And I think with respect to your question about tryptophan, what's worth noting is that tryptophan is one of the rarest amino acids. Relatively few tryptophan residues in a given protein. However, tryptophan is also the largest amino acid, so it's kind of special, and so it's an interesting observation, but there's no specific, rational reason that we understand at this point. Yeah, someone else. I'm going to let Jean call it.
Suzanne?
Yeah.
Yeah, I just wanted to give a a funny comment regarding the Y chromosomes. That one of my professors in medical school, he'd say to all the guys, there's no wonder that women are so superior to us. We're all missing half a chromosome.
And that we can actually say is evolutionarily true. The Y chromosome represents an evolutionary vestige of the X chromosome, and there's a little part of the X chromosome that is called a little part of the Y chromosome, I mean, that's called the pseudo X chromosome, and that allows the Y chromosome and the X chromosome to pair during the meiosis, and so that sort of allows one set of chromosomes to go one way in a man to make the female sperm, and another set of chromosomes to go the other way to make the male sperm. yeah, no, so that's interesting, I think.
In the chat there, Walter has a question. Will you be covering sense sequences versus nonsense sequences or spacer sequences?
Yeah. Let me go on and talk about the genome, because the genome is full of lots of what we call non-coding RNA or non-coding DNA rather, and RNA molecules can be divided as coding molecules, that is messenger RNAs, and non-coding RNAs, that's ones that don't code for proteins. And so we do talk about spacer sequences. But I'm not quite sure what the questions focus on, because there's some different things here. We can talk about antisense and sense being the two different complementary types of molecules. So you can have the strands of DNA can be thought of as sense strands coding for messenger RNA, and the antisense strands that are complementary to it. So there are a number of different ways of classifying sequences. I'll talk a little bit about that as we go on. Yeah. Any other questions? So I see the hour is about up and people have to leave, but we're going to get into the Genome Project itself, and I'm going to sort of accelerate. We're going to talk about in very broad terms here. I want to kind of talk about the basics of what people found out when they set out to sequence the genome, and a little bit about sort of the latest developments that I think are sort of very, very interesting and exciting. Should I go on then? Jean, we're okay?
Sure, go for it.
Yeah. Okay. Back to the screen share. Let's see. Okay, now it take me a second to get these slides operating. All right. So does everyone see this now? I just want to bring us into the 70s, 80s, 90s, and 21st century rather quickly. In the 70s, so everything I've talked about so far, really, I've sort of jumped ahead to talk about the details of protein structure, but everything really was back in the 50s and 60s. In the early 70s, they discovered a whole class of enzymes called restriction enzymes that cut DNA at specific sites, that has a specific sequence of bases of sometimes as few as 4 bases, sometimes 6 or 8 bases. These restriction enzymes isolated from bacteria would cut DNA at specific sites. And that sort of allowed for the next advance a couple years later of a technique called molecular cloning, which allowed people to take specific sequences of DNA and combine them with another type of DNA, typically either a virus DNA that could replicate autonomously or other types of DNA known as plasmids that could replicate by themselves when put into a host cell. And so molecular cloning together with restriction enzymes allowed you to take specific fragments of DNA and copy them into and also to express them. That is to take a copy of DNA containing a particular gene and getting it to express the protein that that gene coded for. And so molecular cloning provides a technique in which you could generate a lot of very specific DNA molecules, but also a lot of specific RNA and protein molecules. And it's now a means of producing many protein drugs, including insulin. So it's interesting that insulin was one of the first proteins that people used to study the very basic structure of proteins as a class of biomolecules. And it was also one of the first products of recombinant DNA technology, actually the first commercial product of molecular cloning. And then finally in 1977, we had the advent of rapid DNA sequencing. And up until 1976, when the technique began to be disseminated, because this was a technique invented by none other than Frederick Sanger, the man who had first sequenced proteins. And he was very generous with his methods, so that by the time he published his work in 77, there were people that had already been using it. And it proved to be a very convenient way of sequencing clone genes and viruses. And people began to be actually able to study DNA at the level of sequence. And so here we have, once again, Frederick Sanger looking at the results of one of his sequencing experiments. And what was so amazing about this method that Sanger worked out was that it was very adaptable, and refinements were quickly developed. And in 1990, the first automated DNA sequencer was introduced. And coincidentally, of course, that was the first year where the Human Genome Project formally began. And so it was only with the use of this automated Sanger sequencing that people were able to make a run at studying millions and billions of bases of DNA. So the other impetus, not only was there a method and developing methods for sequencing DNA, but beginning in the late 70s, people had worked out very tedious techniques for mapping and identifying genes using the techniques I just described, the restriction enzymes and the molecular cloning techniques, and of course, sequencing techniques. So people were able to, with a great deal of effort, a lot of cooperation between labs, were able to work out the basis, the molecular basis for a couple diseases that had been very puzzling. Cystic fibrosis is a fairly common genetic disease, not quite as common as the sickle cell disease is among African populations, but cystic fibrosis is quite common in European populations. And one in a few thousand people suffer from cystic fibrosis. And people had no idea what the underlying basis was, but it was clear from genetics that it was a genetic disease. And so a couple labs played a major role in identifying cystic fibrosis based primarily just on the studying DNA samples from related individuals, that is, individuals in a family where some had the disease and other people didn't. And this was called linkage analysis. And it was also applied to Huntington's disease, which is a rare disease, but a particularly devastating one. It's sometimes known as Woody-Guthrie's disease, because whereas cystic fibrosis occurs in very early childhood, Huntington's disease occurs in middle age, but it's a very debilitating disease, both physically and mentally, and eventually leads to death. There are now drugs for cystic fibrosis. There is no cure so far for Huntington's disease, although there are some things under testing. But this success in mapping and identifying the genetic basis for diseases was a powerful impetus for the Human Genome Project. Now we talk about there being something on the order of 7,000 human genetic diseases representing perhaps 5,000 genes. We'll see what this means. But at the time that they began the Human Genome Project, there were only a handful of genetic diseases where the disease gene had actually been recognized. Now we recognize the genetic underpinnings of the majority of those 7,000 diseases, each one of which may be quite rare, but collectively it seems like essentially 10% of people have some genetic anomaly. So in 1990, it started under the very able direction of James Watson, who took strong positions on a number of important topics in strategizing. And the first several years of the Human Genome Project were devoted to technical development and mapping, using techniques I've referred to, but not really described. But really an important part of this early phase of the Genome Project was that different groups undertook sequencing of a number of small genome pilot projects. And so entire genomes of bacteria were sequenced beginning in 1995, 5 years after the start. The yeast genome, which is almost as small as a bacteria's genome, was sequenced, and that was important because the yeast is actually representative of higher organisms, of nucleated cells, but a rather simple one. And then you have worms and flies, which are well-developed genetic models, where it's using genetics, it's possible to discover the function of a particular disease, where it's very, very difficult to do meaningful genetics directly in humans without having a good sequence of DNA. So worms had a genome. Worms are a very tiny animal, a millimeter or two long, and they have no more than 1,000 cells in them, and they have a genome that's about 1/30th the size of our genome and about 20,000 genes. The fly has a genome that's a little larger and maybe 14,000 genes. So there's not perhaps a good correlation between the size of the genome and the number of genes. And so there's a lot of speculation about the size of the human genome. And when it was finally published, beginning in 2001, but finally in 2003, people concluded to their amazement that the human had only about 23,000 genes. And this was quite a reduction because earlier estimates had been that humans had maybe 100,000 genes. And this picture here just shows linear displays of all 24 chromosomes, that is the 23 pairs with an X and Y chromosome making the 23rd pair. So there are 24 individual chromosomes in the body. And what this slide is showing is the regions where the DNA is either highly repetitive or gapped or both, because the genome that was published in 2003 was only about 90% complete, contained over 300 gaps in it. And it's taken the last 20 years to fill in those gaps. But in the last 2 years, each of these chromosomes, each of these individual chromosomes has been sequenced from one end to the next. And as I just said, the last of the chromosomes was published. The sequence of the entire Y chromosome was published last summer. And the problem is that the protein coding regions, the regions that I've focused on so far, in fact, represent less than 2% of the entire length of the genome. And all the other sequences, a lot of them consist of this highly repetitive DNA that is sometimes referred to as junk DNA. And let me just pause for a second while I do some little house cleaning on my computer. It turns out that this estimate of 23,000 genes is just that, and that the most recent estimates are that there's somewhat less than 20,000 genes in the human genome, which talks in terms of gene numbers. We're right on the level of the lowly worm, this nematode worm that contains only 1,000 cells. And it turns out that probably the difference here is in regulation, that many human genes code for more than one protein. They can make more than one messenger RNA, and that the regulation of this genome, of the human genome, is perhaps far more complex in some ways than that of other animals that have as many or more genes, and some rather simple organisms have far more DNA in them. But the regulation of the genes is perhaps the clue. And regulation, it turns out, is relying both on other proteins that bind to the DNA, but it also depends on many RNA molecules, both large and small, that interact with the DNA and interact with proteins and interact with other DNA molecules. So in fact, as people study the RNA within given cells, they've discovered tens of thousands of non-coding RNAs, which fall into a number of discrete classes. And these are being intensively studied now. They were first recognized in the early 2000s, or the late 1990s, when it suddenly became clear that there was a whole level of small RNA molecules that carried out specific and subtle regulatory functions that had been totally overlooked. And I think the identification of these non-coding RNAs is one of the most valuable and also challenging problems of understanding the genome. So that kind of summarizes a lot of things up. This just shows a lab in England that decided to print out the entire genome, just again to emphasize how vast it is. These books are color-coded according to the staining of the chromosomes of the DNA in that first slide I showed you at the beginning. So there are 130 volumes here with a lot of small print on each page, totaling a total of over 3 billion letters of DNA. And there are a couple copies of this, and I noticed that another university in England had sort of copycatted the people at Leicester University and made their own set. So just to make some general remarks here before we tie this up, the completed genome has, beginning in even the late 1990s, the genome sequence has become an indispensable reference for research, and now has many clinical applications, ones that are particularly important because the genetics turns out to be a really critical way for studying cancer and for understanding differences between different types of tumors and following their response to therapies. The function of many genes is still poorly understood. So we know we have about 20,000 genes in our genome. We do not know what all the proteins that those genes code for do, but the way in which we're understanding the function of many proteins is to compare them with related proteins found in many different species. So compiling genetic information from many different types of organisms and from many individuals has been important for understanding the function of specific genes. And almost immediately, as the genome was being finished in 2003, new technologies came online unrelated to Sanger sequence. Some of them, in fact, were modifications of Sanger sequencing, but many of them totally novel approaches. And so DNA and RNA sequencing has become extraordinarily faster and cheaper in the last 2 decades. And so now you can get your genome sequenced in a couple of days for a couple $100. The quality of that sequence depends on just how thoroughly you sequence it. It gets more expensive as you try to get more and more precise, perhaps, but you can get. We now know the sequence of tens of thousands, hundreds of thousands of bacteria, thousands of different animals, plants, and fungi have all been sequenced now. But despite the mass of this information that's pouring out of laboratories, understanding many levels of regulation is still the major challenge that faces us. And I just a quick slide, just sort of summarizing some of the aspects of regulation in the human genome. Is we have a level called packaging, where proteins bound to the DNA within the chromosome can interact with the DNA in a number of different ways. And this contributes to the accessibility of genes to other factors, which can turn on and off RNA transcription of a particular DNA. It's sort of hard to recognize sequences within a gene or outside of a gene that regulate that gene, because regulatory elements are often located tens of thousands of bases away from the gene of interest. And so it takes a considerable amount of effort and genetics to understand how a set of genes is regulated. And then finally, how I've mentioned that one gene can code for many different proteins. And this reflects the fact that in higher organisms, most coding regions are divided into multiple pieces, and that non-coding segments called introns have to be removed from the first RNA copy of a gene before that RNA becomes a functional messenger RNA and can be translated into protein via the ribosomes. Moving up to the present time, in the year 2012, 2 biochemists, Jennifer Doudna at Berkeley in California and Emmanuel Charpentier, formed a short-lived but very productive collaboration that quickly won them the Nobel Prize in 2020. They were able to develop a system that could specifically edit genes in almost every type of organism. And this system that they developed is known as CRISPR, which is a palindrome. I mean, what's the word I'm looking for? It's an acronym for a certain type of sequence that was first noted as bacterial genomes began to be sequenced. So as bacterial genomes began to pour out of sequencers in the mid-1990s, people noticed some strange sequences which had not been noted by geneticists or other people studying the same bacteria for many decades. And these sequences turned out to code for a type of bacterial immune system in which bacteria can slice up a piece of DNA from an invading virus and then store it so that it recognizes that virus, assuming the cell survives the first viral attack. And within that system, there was a method for editing human cells. And so this system was engineered by Doudna and Charpentier into a protein called Cas9, which bound to what's called a guide RNA that is targeted for a very specific site within any given genome. And so the Cas9 is cuts the DNA in a specific way, and that sets up the process of editing in that site that's being attacked. And so this turns out to be an extraordinarily versatile system that can be scaled to use in large-scale research of looking for genes and studying gene functions, but it can also be used for editing genes within this chromosomes of an organism. And so finally, I want to talk about the recent success in using this type of genetic technique to cure sickle cell anemia. And the method that has gained sort of has won the first round of approval from the FDA and has been successfully used in dozens of cases now with relatively few side effects, because it's a fairly involved procedure. I just want to give you a little bit of ideas of the complexity that we're dealing with when trying to manipulate human genes. This shows what's known as the beta globin locus. And this is a site within the human chromosome that codes for the beta chain in the hemoglobin protein. And it actually contains 5 copies of genes, of related genes for chains that are used in hemoglobin, starting with the epsilon gene here, which codes for an embryonic form of the beta chain, which is replaced by a fetal form coded by 2 copies of this gamma globin gene. And then that's followed by 2 copies of the delta and the beta gene itself, which code for adult forms of hemoglobin. So hemoglobin, looking at the human genome, we discovered that globin is more complex than Pauling had any reason to realize. The beta form is by far the majority of forms, so people could have been forgiven for overlooking the delta form for some years. And interestingly, the fetal form can substitute for the adult form. So typically the embryonic form is expressed in very early development. It's turned off and the gamma forms are turned on, and the gamma forms are replaced by adult forms in early infancy, in the first few months of postnatal development. So babies are born expressing hemoglobin that contains alpha chains and gamma chains rather than alpha chains and beta chains. And there is a condition called persistence of human fetal hemoglobin, which is a genetic condition, but it's not really a disease. People with it actually are fairly normal. And it turns out that the expression of fetal hemoglobin can counter the sickling effects of the abnormal beta chain in sickle cell anemia. So this little line here indicates we have an abnormal gene. And all of this array of genes is controlled by a distant regulatory site on the DNA called the LCR site. And this interacts with these different genes in turn. So the LCR here is shown activating the adult form. And it does it after another regulatory site with 2 proteins, actually a large number of proteins bound, but just 2 are indicated here, actually act to repress the fetal form. So the LCR is a positive regulator. This complex here is a negative regulator. And the way in which this the mutated sickle cell locus was modified was people actually attacked this repressor gene called BCL11A. Gene nomenclature gets very complicated now. And whoa, something just happened. Are we still online? Yes. Okay. Let me go back because I'm not sure what just happened then. See if I can get back to where I was. I just have a message here saying that my screen share has closed, and it looks like that whole window closed. So I'm going to have to go back to my, see if I can get back to my slide. I'm almost finished. So stick with me for another minute. Here we go. Oops. Can everyone see that? Jean, are we back
Yes.
online? Good.
Yes.
Thank you. Not sure what happened there. So this is a very indirect approach, but this is the first application of CRISPR, the gene editing technique that was developed, first pioneered by Doudna and Charpentier. This is the first application of CRISPR to editing human genes, and it was just approved by the FDA in December for sickle cell anemia, and in January they followed up by approving it for another hemoglobin disease called beta thalassemia, and in both of those diseases we have either absent or defective forms of beta globin, and so this treatment has the effect of turning on the fetal form of hemoglobin, which counters or replaces the sickle cell form, and it's been very successful in more than 95% of individuals in the testing over a period of 2 or 3 years, and gene therapy has turned out to be difficult, but this has worked, and there's been a series of articles. Many in the Boston Globe, because this was carried out by Vertex Pharmaceuticals, which is a Cambridge, Massachusetts company in collaboration with CRISPR Therapeutics, a Swiss biotech company. There are other companies, Bluebird Bio, another Cambridge company, another Massachusetts company anyway, is also working on a gene therapy, sickle cell anemia. And so I think it's very appropriate that sickle cell disease, which where Pauling's work, very basic work done back more than 70 years ago, 75 years ago now, proved to be a really pioneering bit of work that have established some basic principles of genetic disease and protein biochemistry and also genetic inheritance. As that disease has now come full circle. It's taken more than 70 years to come up with a good solution to it. There were a number of treatments that have been developed over the years, but relatively frustratingly few successful treatments for sickle cell disease. This is a very common genetic disease in African populations, but it's also found in other populations from the Middle East and from India. Evolutionarily, it provides protection against malaria, which is a major killer throughout the world today. Millions of people die from malaria every year. Sickle cell has persisted because people with the sickle cell gene, a single gene, actually show a high level of resistance to malaria. But it's a mixed inheritance in today's society. So Casgevy is a full treatment. It's very similar to the treatment for many bone cancers, where immune cells are removed from the bone marrow and then edited and then replaced so that the person will express the genes in their bone marrow that will lead to new and edited red blood cells. So that concludes my talk. This is the story I have for you today. How early work in decoding genes has kind of foreshadowed the modern work, and we're confronted, as research often is, we're confronted with as many questions in 2024 as scientists were in 1953 when they first began to unravel the nature of the gene and human genetics. So thank you.
Thank you. It's great.
Thanks for.
Thank you, Steve. That time just flew by.
Well, thank you, Paul. It's good to see so many of you again. Are there
Thank you. That was very informative.
Good. Okay. Well.